You know, there's something almost paradoxical about sleep. We spend roughly a third of our entire lives in this state, unconscious, vulnerable, seemingly doing nothing at all. And yet, if you were to ask any neuroscientist, any sleep researcher, anyone who has spent years studying the intricate workings of the human brain, they would tell you that sleep is anything but passive. It's not a shutdown. It's not a simple rest period where the lights go off and everything goes quiet. Sleep is an extraordinarily active, meticulously orchestrated biological process that is absolutely fundamental to who we are, how we think, how we feel, and how our bodies function at the most basic cellular level. And here's the thing that I find so fascinating. For most of human history, we didn't really understand what was happening during those hours of darkness. Sleep was a mystery, a black box. We knew we needed it. That much was obvious from the profound consequences of going without it. But the why and the how remained elusive. It's only in recent decades, really in the last 20 or 30 years, that science has begun to pull back the curtain and reveal the hidden biology of sleep. And what we've discovered is nothing short of remarkable. We've learned that sleep is when the brain performs some of its most critical maintenance work, when memories are consolidated and strengthened, when emotions are processed and regulated, when the very architecture of our neural networks is refined and optimized. We've learned that sleep is when the brain quite literally cleans itself, flushing out toxic waste products that accumulate during our waking hours. We've learned that the quality and structure of our sleep can influence our risk for some of the most devastating diseases of aging, from Alzheimer's to Parkinson's to diabetes and heart disease. So in this video, I want to take you on a journey into the hidden world of sleep. We're going to explore what actually happens in your brain and body during those hours when you're lying still, eyes closed, drifting through different stages of consciousness. We're going to talk about the elegant regulatory systems that govern when you feel sleepy and when you feel alert, about the intricate architecture of sleep stages that cycle throughout the night, about the master clocks and peripheral timekeepers that orchestrate your body's daily rhythms. We're going to dive deep into one of the most exciting recent discoveries in neuroscience, the Glymphatic system, a brain-wide waste clearance network that operates primarily during sleep and may hold the key to understanding how sleep protects against neurodegeneration. And we're going to connect all of this biology to practical implications for your health, your cognitive performance, your emotional well-being, and your long-term brain health. This is going to be a detailed exploration because sleep deserves that level of attention. It's not a simple topic, and don't want to oversimplify it, but I also want to make it accessible, to paint a picture that helps you understand not just the what, but the why. Why sleep matters so profoundly, and why protecting and prioritizing your sleep might be one of the most important things you can do for your brain and your body. So settle in, get comfortable, and let's begin this journey into the hidden biology of sleep. The two pillars of sleep. Regulation, understanding what makes you sleepy. Let's start at the very beginning with a fundamental question. What makes you feel sleepy? What determines when you're ready to fall asleep and when you're ready to wake up? It might seem like a simple question, but the answer reveals an elegant system of biological regulation that has been refined over millions of years of evolution. The timing, the duration, and even the intensity of your sleep are governed by two distinct but beautifully interconnected processes. Sleep scientists call this the two-process model, and it was first proposed by a researcher named Alexander Borbet more than 40 years ago. Despite its age, this model remains the cornerstone of our understanding of sleep regulation. The first process is called Process S, and it represents your homeostatic sleep drive. Think of it as sleep pressure, a biological force that builds up progressively the longer you stay awake. It's like an hourglass slowly filling with sand. Every minute you're awake, every thought you think, every movement you make, every sensory experience you process, all of this activity is adding to that accumulating pressure. And the longer you go without sleep, the more powerful that drive becomes, until eventually it becomes almost irresistible. You've probably experienced this yourself if you've ever stayed up all night or pushed through extreme fatigue. There comes a point where your body is simply demanding sleep, where your eyelids feel impossibly heavy, where your attention starts to drift and your thoughts become foggy. That's Process S at work, that homeostatic drive reaching a critical threshold. But what's actually happening at the molecular level to create this sleep pressure? The answer lies in a small molecule called adenosine. Adenosine is a neuromodulator, a chemical messenger in the brain, and it's produced as a natural byproduct of cellular energy consumption. Every cell in your body, and especially the incredibly active neurons in your brain, runs on a molecule called ATP, adenosine triphosphate. ATP is the universal energy currency of life. When your neurons fire, when they communicate with each other, when they perform the countless computations that underlie every aspect of your conscious experience, they're breaking down ATP to release energy. And when ATP is broken down, one of the byproducts is adenosine. So as you go through your day, as your brain is working, thinking, processing, learning, adenosine is steadily accumulating in the extracellular space, the fluid-filled gaps between your brain cells. This accumulation is a direct signal of your brain's metabolic activity, a measure of how much energy you've been burning. And here's where it gets really interesting. Adenosine doesn't just sit there passively. It actively promotes sleep by binding to specific receptors on neurons, particularly the A1 and A2A receptor subtypes. When adenosine binds to these receptors, it has two complementary effects. First, it inhibits the activity of wake-promoting neurons, the cells in your brain that keep you alert and conscious. These wake-promoting neurons are located in several key arousal centers, including the basal forebrain, the hypothalamus, and the brainstem. Second, adenosine simultaneously excites sleep-promoting neurons, particularly those in a region called the ventrolateral preoptic area, or VLPO, and the median preoptic nucleus, or MNPO. These sleep-promoting nuclei are absolutely critical. They're rich in a neurotransmitter called GABA, gamma-aminobutyric acid, which is the brain's primary inhibitory signal. When the VLPO and MNPO become active, they essentially act as a master switch for sleep, sending out widespread inhibitory signals that suppress the brain's arousal systems. It's like they're turning down the volume on wakefulness, allowing sleep to take over. And the trigger for this switch is the rising level of adenosine, that molecular marker of how long you've been awake and how much energy your brain has expended. Now, here's the beautiful part. During sleep, especially during the deep, slow-wave stages that we'll talk about in detail later, your brain's metabolic rate decreases. The frenetic activity of wakefulness slows down. And in this quieter state, your brain can clear away the accumulated adenosine, breaking it down and reconverting it back into ATP, replenishing your energy stores. This is why sleep is restorative. It's not just about resting, it's about actively clearing the molecular debt that has built up during wakefulness. When you wake up after a good night's sleep, your adenosine levels are low, your sleep pressure has been dissipated, and you feel refreshed and alert, ready to start the cycle all over again. Strongly supported, according to multiple studies on adenosine's role in sleep homeostasis, including research published in PMC and Nature Communications. But here's the thing. Process S, this homeostatic sleep drive, doesn't work in isolation. If it did, you would just get progressively sleepier throughout the day until you eventually collapsed into sleep. And then you'd wake up whenever your adenosine levels had been sufficiently cleared, regardless of the time of day or night. But that's not how it works. You don't feel equally sleepy at all times of the day, even if you've been awake for the same number of hours. There are times when you feel naturally alert, even if you've been up for a while, and times when you feel sleepy, even if you haven't been awake for that long. This is where the second process comes in. Process C, the circadian process. Process C is governed by your body's internal biological clock, a self-sustaining, approximately 24-hour rhythm that is independent of how long you've been awake. This clock is located in a tiny cluster of neurons in your brain called the suprachiasmatic nucleus, or SCN, which sits in the hypothalamus, right above the point where your optic nerves cross. The SCN is your master circadian pacemaker, and it generates a powerful rhythmic signal that promotes wakefulness during the day and allows for sleep at night. Think of it as a wave of alertness that rises and falls over the course of 24 hours. Here's how it works in concert with Process S. Throughout the day, as your homeostatic sleep pressure is building up due to rising adenosine, your SCN is simultaneously generating a strong, wake-promoting signal. This circadian drive for wakefulness effectively counteracts the rising sleep pressure, allowing you to stay alert and functional even as adenosine accumulates. It's like the SCN is pushing back against the weight of that hourglass, keeping you upright and awake. But as evening approaches, the SCN's wake-promoting signal begins to wane. It starts to open a gate, creating a window of opportunity for sleep to occur. And this window coincides with the peak of your homeostatic sleep pressure. So you have two forces converging. The sleep drive from Process S is at its highest, and the wake drive from Process C is at its lowest. This is when you feel most ready for sleep, when falling asleep feels natural and easy. The SCN also controls the production of a hormone that you've probably heard of, melatonin. Melatonin is produced by the pineal gland, a small endocrine gland deep in your brain. During the day, when light is present, the SCN actively inhibits the pineal gland, keeping melatonin levels low. But as darkness falls, the SCN lifts this inhibition and melatonin synthesis and secretion ramp up. Melatonin doesn't directly cause sleep. It's not a sedative in the traditional sense, but it facilitates the transition to sleep. It enhances the activity of those sleep-promoting neurons in the VLPO and reduces the activity of arousal-promoting brain regions. It's a signal to your body that night has arrived and that it's time to prepare for sleep. Strongly supported, according to research on the suprachiasmatic nucleus and circadian regulation published in PMC and Nature Reviews, Neuroscience. So you can see how elegant this system is. Process S and Process C work together, two complementary mechanisms that ensure sleep occurs at the right time and for the right duration. The homeostatic drive ensures that you get enough sleep to clear your metabolic debt, while the circadian drive ensures that this sleep happens at a biologically appropriate time, aligned with the external cycle of day and night. The greatest propensity for sleep. occurs when these two processes are in sync, when your sleep pressure is high and your circadian drive for wakefulness is low. This dual control system highlights a crucial point. Sleep is not a passive state that just happens when you're tired. It's a precisely regulated biological imperative, driven by complex neuronal circuits and molecular mechanisms that have been honed by evolution to keep you healthy and functioning optimally. The architecture of sleep, a journey through depth, night now. That we understand what makes you sleepy, let's talk about what actually happens once you fall asleep. Because sleep is not a single, uniform state. It's not like flipping a switch and entering a static condition of unconsciousness that lasts until morning. Sleep is dynamic, structured, and cyclical. It's a journey through distinct stages, each with its own unique characteristics, its own patterns of brain activity, and its own specific functions. This organization is called sleep architecture, and understanding it is key to appreciating why both the quantity and the quality of sleep matter so much. When you fall asleep, you don't immediately plunge into the deepest stages. You enter gradually, moving through progressively deeper levels of what's called non-rapid eye movement sleep, or NREM sleep. NREM sleep is divided into 3 stages, which we'll call N1, N2, and N3. Stage N1 is the lightest phase, that brief transitional state between wakefulness and sleep. It usually lasts only a few minutes. During N1, your muscle tone begins to relax, your heart rate starts to slow, and your brain activity shifts. If you were to measure your brain waves with an EEG, an electroencephalogram, you would see a transition from the rhythmic alpha waves that characterize relaxed wakefulness to slower theta waves. You're drifting, hovering on the edge of consciousness. You might experience those sudden jerks or twitches, called hypnic jerks, as your muscles relax. And you can be easily awakened from this stage. A small noise or a gentle touch might bring you right back to full wakefulness. After a few minutes in N1, you transition into stage N2, which is a more stable and deeper phase of sleep. N2 accounts for the largest proportion of your total sleep time, typically around 45 to 55% in adults. This is where sleep really begins to take hold. Your body temperature drops. Heart rate and breathing become more regular and stable, and your brain starts to exhibit some very specific and fascinating electrical patterns. On an EEG, N2 sleep is characterized by 2 distinctive waveforms: sleep spindles and K-complexes. Sleep spindles are brief bursts of high-frequency brain activity, rapid oscillations that last for a second or two. They look like little spindles on the EEG trace, hence the name. These spindles are thought to play a crucial role in memory consolidation, helping to transfer information from short-term to long-term storage. They may also serve a protective function, helping to keep you asleep by suppressing your brain's responses to external stimuli. K-complexes, on the other hand, are large, slow waves, sudden, sharp peaks and troughs in the EEG. They can occur spontaneously or in response to external sounds or disturbances, and they're believed to help maintain sleep by preventing arousal. Together, these features make N2 a stable, restorative stage where your body and brain are settling into deeper rest. Strongly supported, according to research on sleep stages published in Sleep Foundation and NCBI Bookshelf. But the deepest and most physically restorative stage of sleep is stage N3, also known as slow wave sleep, or SWS, or sometimes just deep sleep. This is where the magic really happens. N3 is defined by the presence of high-amplitude, low-frequency delta waves on the EEG. These are slow, powerful oscillations that sweep across the brain, a synchronized rhythm that reflects a profound state of neural quietude. During N3, your brain's metabolic rate drops significantly. Your heart rate and breathing slow to their lowest levels of the night. Your blood pressure decreases, and it becomes very difficult to wake you up. If someone does manage to rouse you from deep sleep, you'll likely experience a period of disorientation and grogginess called sleep inertia, where your cognitive functions are impaired and you feel almost drunk with sleepiness. But while you might seem almost comatose during N3, your body is anything but inactive. This is when some of the most critical restorative processes occur. Deep sleep is the primary period for the release of growth hormone, a hormone that is essential for cellular repair, for building and maintaining bone density, for preserving lean muscle mass, and for overall physical recovery. It's also a crucial time for immune system reinforcement. Your body ramps up the production of cytokines and other immune factors that help you fight off infections and heals from injuries. And, as we'll explore in great detail later, N3 is the stage when the brain's waste clearance system, the glymphatic system, is most active, flushing out toxic metabolic byproducts that have accumulated during wakefulness. Deep sleep is, in many ways, the foundation of physical restoration. Strongly supported, according to research on slow-wave sleep published in PMC and Sleep Foundation. After progressing through these NREM stages, your brain doesn't just stay in deep sleep for the rest of the night. Instead, it transitions into a completely different state, REM sleep, which stands for Rapid Eye Movement Sleep. REM sleep is paradoxical. If you were to look at an EEG during REM, you would see brain activity that looks remarkably similar to wakefulness. The brain is highly active, almost as active as when you're awake and alert. But your body is in a state of near-complete paralysis. Your voluntary muscles, except for your diaphragm and the muscles that control your eyes, are essentially shut down, a condition called atonia. This paralysis is a protective mechanism, a safety feature that prevents you from physically acting out the vivid, often bizarre, narrative-rich dreams that are the hallmark of REM sleep. During REM, your eyes dart rapidly back and forth beneath your closed eyelids, hence the name. Your heart rate and breathing become variable and irregular, sometimes speeding up, sometimes slowing down, reflecting the emotional content of your dreams. And the neurochemical environment of your brain changes dramatically. REM sleep is characterized by high levels of acetylcholine, a neurotransmitter that is crucial for learning and memory, and by a suppression of monoamines like norepinephrine and serotonin, which are typically associated with wakefulness and focused attention. This unique neurochemical cocktail is believed to be essential for several critical functions: the consolidation of procedural memories, the skills and habits that you learn through practice, the processing and regulation of emotions, and even creativity and problem-solving. There's a reason why people often say they need to sleep on it when facing a difficult decision or a creative challenge. REM sleep provides a space for your brain to make novel connections, to integrate new information with old, to work through emotional experiences in a safe, neurochemically modulated environment. Strongly supported, according to research on REM sleep published in PMC and sleep medicine clinics. Now here's where the architecture of sleep gets really interesting. These stages don't just occur once, and then you're done. They cycle. Throughout the night, you move through repeated cycles of NREM and REM sleep, each cycle lasting approximately 90 to 120 minutes. A healthy adult typically experiences 4 to 6 of these cycles per night. But the composition of these cycles changes as the night progresses, and this temporal organization has profound implications for what happens if you cut your sleep short. In the first half of the night, your sleep is dominated by deep, slow-wave sleep. The N3 stages are longest and most intense during the first few cycles. This makes sense from a biological perspective. You've been awake all day, your homeostatic sleep pressure is at its peak, and your brain prioritizes the deep, restorative sleep that is needed to dissipate that pressure and perform critical physical maintenance. This is when your brain is doing its heaviest lifting in terms of waste clearance, immune function, and hormonal regulation. This front-loading of deep sleep is essential for feeling physically rested and recovered. But as the night goes on, the amount of N3 sleep decreases, and the amount of REM sleep increases. By the second half of the night, your sleep cycles are characterized by longer and longer periods of REM, interspersed with lighter N2 sleep. The final REM period of the night, the one that occurs just before you wake up, can last up to an hour or even longer. This is when you're most likely to have those long, complex, emotionally vivid dreams that you might remember upon waking. This changing architecture has critical, practical implications. If you go to bed late, cutting into the early hours of your sleep period, you're disproportionately losing deep, slow-wave sleep. You're missing out on that crucial window when your brain is clearing waste, when your immune system is being reinforced, when growth hormone is being released. You might still get some sleep, but you're not getting the full restorative benefit of those early, deep sleep cycles. Conversely, if you wake up too early, if you cut your sleep short by setting an alarm that goes off before you've completed your natural sleep cycles, you're primarily depriving yourself of REM sleep. You're losing that critical time for emotional processing, for memory consolidation, for creativity and problem solving. And this can have significant consequences for your mood, your emotional regulation, your ability to learn and remember new information, and your overall cognitive performance. Strongly supported, according to research on sleep architecture published in Sleep Foundation and NCB iBookshelf. So you can see why both the quantity and the quality of sleep matter. It's not enough to just spend a certain number of hours in bed. You need to allow your brain to move through complete, uninterrupted cycles, to experience the full spectrum of sleep stages in their natural proportions. This is why consistent sleep schedules, adequate sleep duration, and minimizing disruptions are so important. Your brain needs that full architectural journey from the light stages of N1 and N2 through the deep restoration of N3 and into the cognitive and emotional processing of REM, cycling through these stages multiple times across the night. This is how sleep does its work. This is how it keeps you healthy, sharp, and resilient. The master clock and the orchestra of peripheral timekeepers. We've talked about the homeostatic drive for sleep and the circadian rhythm that governs the timing of sleep and wakefulness. But let's zoom in on that circadian system, because it's far more intricate and far-reaching than you might imagine. Your body doesn't just have one clock. It has a hierarchical network of clocks, a master timekeeper, and a vast orchestra of peripheral timekeepers, all working in concert to coordinate your physiology with the 24-hour cycle of day and night. At the top of this hierarchy sits the suprachiasmatic nucleus, the SCN, that tiny cluster of about 10,000 neurons in your hypothalamus. The SCN is your master circadian pacemaker, and its timekeeping ability is truly remarkable. It's generated at the cellular level through a series of self-sustaining molecular feedback loops, a genetic clockwork that operates within each individual neuron. Here's how it works. There are core clock genes with names like clock and BMAL1 that drive the expression of other clock genes, including period, or PER, and cryptochrome, or CRY. The proteins produced by these PER and CRY genes accumulate in the cell's cytoplasm over the course of several hours. Eventually, they reach a critical concentration and they re-enter the nucleus, where they inhibit the activity of CLOCK and BMAL1, essentially turning off their own transcription. This creates a negative feedback loop. As the levels of PER and CRY proteins decline because they're no longer being produced, the inhibition on SLOC and BMAL1 is lifted, and the cycle starts all over again. This entire loop takes approximately 24 hours to complete, and it forms the molecular basis of the circadian rhythm within each SCN neuron. The collective synchronized output of all these neurons generates a robust, body-wide circadian signal, strongly supported, according to research on the suprachiasmatic nucleus published in PMC and Nature. Reviews Neuroscience. Now, this internal clock is self-sustaining. Even if you were to isolate the SCN in a dish in a laboratory, it would continue to tick away with a roughly 24-hour rhythm. But in the real world, this clock needs to be synchronized or entrained to the external environment. Otherwise, it would slowly drift out of alignment with the actual day-night cycle, because the internal period is usually not exactly 24 hours. It's often a bit longer, maybe 24.2 or 24.3 hours. So how does the SCN stay in sync with the outside world? The answer is light. Light is the primary environmental cue, the primary Zeitgeber, a German word meaning time giver, for the circadian system. The SCN receives direct information about light from the retina via a specialized neural pathway called the retinohypothalamic tract, or RHT. This input doesn't come from the rods and cones that you use for vision. It comes from a unique class of retinal ganglion cells that contain a photopigment called melanopsin. These cells are intrinsically photosensitive, meaning they can detect light on their own, and they're particularly sensitive to blue wavelengths. When light strikes these melanopsin-containing cells, they send a signal directly to the SCN, and the SCN uses this information to reset its internal clock every day. The timing of light exposure is crucial. Light exposure in the morning, shortly after you wake up, causes a phase advance, shifting your internal clock earlier. This helps you wake up earlier the next day. Light exposure in the evening, on the other hand, causes a phase delay, shifting your clock later, making you want to stay up later and wake up later. This is why exposure to bright light, especially blue-rich light from screens in the hours before bedtime, can be so disruptive. It's sending a signal to your SCN that it's still daytime, that it's not yet time to prepare for sleep, and this delays the onset of melatonin production and makes it harder to fall asleep at your desired time. Strongly supported, according to research on circadian rhythms and light entrainment published in PMC and Frontiers in Neural Circuits. The SCN also controls the nightly secretion of melatonin from the pineal gland, as we discussed earlier. During the day, light exposure suppresses melatonin production, reinforcing wakefulness. At night, in the absence of light, melatonin levels rise, facilitating the transition to sleep. This is a beautiful example of how the external environment, the internal clock, and the hormonal system all work together to regulate your sleep-wake cycle. But here's where it gets even more fascinating. The SCN doesn't act alone. It coordinates a vast network of peripheral clocks located in virtually every other cell and organ system throughout your body. Your liver has a clock. Your pancreas has a clock. Your heart, your skeletal muscles, your gut, your adipose tissue, they all have clocks. These peripheral clocks possess the same core molecular machinery as the SCN, the same clock, BMAL1, PER, and CRY genes, the same feedback loops. But they rely on signals from the master clock in the SCN to maintain synchrony with each other and with the external environment. The SCN communicates with these peripheral oscillators through multiple channels, through direct neural projections, through hormonal signals like glucocorticoids, and by controlling body temperature rhythms. When the SCN is functioning properly and is well entrained to the light-dark cycle, all of these peripheral clocks tick along in harmony, each one regulating the timing of specific physiological processes in its respective organ. Your liver clock, for example, regulates the timing of glucose production and lipid metabolism. Your pancreas clock regulates insulin secretion. Your gut clock regulates digestive enzyme production and nutrient absorption. This temporal coordination is essential for metabolic efficiency and overall health, strongly supported, according to research on peripheral clocks published in PMC and BMC Biology. But here's the critical point. While the SCN is almost exclusively entrained by light, these peripheral clocks are highly sensitive to other Zeitgebers, and the most powerful of these is the timing of food intake. When you eat has a profound effect on the phase of your peripheral clocks, particularly in metabolic organs like the liver. Studies have shown that if you restrict food access to specific times of day, you can shift the phase of the liver clock, even if the SCN's light-dark cycle remains unchanged. This creates a potential for internal desynchronization, a mismatch between the central clock and the peripheral clocks. Imagine this scenario. You're a shift worker and you're eating meals in the middle of the night when your SCN is signaling that it's time to sleep. Your SCN is saying, it's nighttime, shut down metabolic processes, but your liver and pancreas are receiving a conflicting signal from the food you're eating, saying, it's time to be active, process nutrients, produce glucose. This internal conflict, this desynchrony, disrupts metabolic processes and is associated with an increased risk for obesity, type 2 diabetes, cardiovascular disease, and even certain cancers. It's one of the reasons why shift work is so detrimental to health. Strongly supported, according to research on circadian misalignment and metabolic health published in PMC and Frontiers in Endocrinology. There's also a concept called social jet lag, which describes the chronic misalignment between your endogenous biological rhythm, your natural chronotype, your internal preference for when you want to sleep and wake, and the demands of your social schedule, like early work or school start times. If you're naturally a late chronotype, a night owl, but you're forced to wake up at 6 a.m. every day for work, you're essentially living in a state of perpetual jet lag. Your internal clock is saying one thing, but your external schedule is demanding another. This forced desynchrony can lead to chronic sleep deprivation, metabolic dysfunction, mood disturbances, and a host of other negative health consequences. The takeaway here is that maintaining alignment, not only with the external light-dark cycle, but also within your body's internal circadian network, is critical for long-term health. This means getting regular, bright light exposure during the day, especially in the morning. It means minimizing light exposure, particularly blue light, in the evening. It means eating your meals at consistent times, ideally during the day when your metabolic organs are primed to process nutrients. And it means, as much as possible, respecting your natural chronotype and aligning your sleep schedule with your internal biological preferences. Your body is a finely tuned orchestra of clocks, and when they're all playing in harmony, you feel good, you function well, and you're setting yourself up for long-term health. But when they're out of sync, when there's discord in the orchestra, that's when problems arise. The brain's nightly cleaning crew, the glymphatic system. Now we come to one of the most exciting and relatively recent discoveries in neuroscience, a finding that has fundamentally changed our understanding of why sleep is so essential for brain health. I'm talking about the glymphatic system, a brain-wide waste clearance network that operates primarily during sleep and plays a critical role in protecting against neurodegenerative diseases. For a long time, it was a mystery how the brain got rid of its metabolic waste. Every cell in your body produces waste products as a byproduct of normal metabolism. In most of your body, these waste products are cleared away by the lymphatic system, a network of vessels that drains fluid and waste from tissues and returns it to the bloodstream for disposal. But the brain doesn't have a traditional lymphatic system. It's separated from the rest of the body by the blood-brain barrier, a highly selective filter that protects the brain from toxins and pathogens in the blood, but also makes it difficult for large molecules to move in and out. So how does the brain clean itself? The answer came in 2012, when a team of researchers led by Maiken Nedergaard at the University of Rochester discovered the glymphatic system. The name is a combination of glia, referring to the glial cells that support neurons, and lymphatic, reflecting its function as a waste clearance system. The glymphatic system is a macroscopic network that facilitates the efficient removal of soluble proteins and metabolic byproducts from the brain's interstitial fluid, the fluid that surrounds brain cells. Here's how it works. Cerebrospinal fluid, or CSF, which is the clear fluid that bathes the brain and spinal cord, is driven into the brain tissue along the outside of penetrating arteries in a space called the para-arterial space, or perivascular space. Think of it like water flowing along the outside of a pipe. Once this CSF enters the brain parenchyma, the actual brain tissue, it mixes with and exchanges with the interstitial fluid, effectively collecting waste solutes that have accumulated in the spaces between cells. This mixture of CSF and waste-laden interstitial fluid is then directed toward the perivenous spaces, the spaces surrounding the brain's veins, and it's ultimately cleared from the brain into meningeal and cervical lymphatic vessels for disposal. Strongly supported, according to research on the glymphatic system published in PMC and Frontiers in Neurology. This process is heavily dependent on a specific type of water channel called aquaporin 4, or AQP4. These channels are densely expressed on the end feet of astrocytes, a type of glial cell that wrap around the brain's blood vessels. AQP4 channels facilitate the rapid movement of water and, by extension, the movement of CSF and interstitial fluid through the brain tissue. Without functional AQP4, the glymphatic system doesn't work properly, and waste clearance is severely impaired. Now, here's the crucial discovery. The glymphatic system is not equally active all the time. It's profoundly state-dependent. During wakefulness, the glymphatic system is largely inactive, operating at a very low level. But during sleep, it becomes dramatically upregulated, increasing its activity by as much as tenfold or more. Research has shown that during sleep, the brain's interstitial space actually expands, increasing in volume by about 60%. This expansion reduces the resistance to fluid flow, allowing for a much more efficient exchange of CSF and interstitial fluid, and a much more effective clearance of waste products. This enhanced clearance is particularly prominent during the deep, slow-wave stages of NREM sleep, during N3. Strongly supported, according to research on the glymphatic system and sleep, published in Science and PMC. So what kind of waste is the glymphatic system clearing? Among the most critical targets are neurotoxic proteins, and two of the most important are beta amyloid and tau. Beta amyloid is a protein fragment that is produced as a normal byproduct of cellular metabolism in the brain. In healthy individuals, it's cleared away efficiently, but if clearance is impaired, beta amyloid can accumulate and aggregate, forming the plaques that are a pathological hallmark of Alzheimer's disease. Tau is another protein that, in its normal form, helps stabilize the internal structure of neurons. But when tau becomes abnormally modified, it can form tangles inside neurons, which are also a hallmark of Alzheimer's and other neurodegenerative diseases. The glymphatic system is a major route for clearing both beta amyloid and tau from the brain, and its activity during sleep is essential for preventing their accumulation. Studies have shown that even a single night of sleep deprivation can lead to a measurable increase in beta amyloid levels in the cerebrospinal fluid of healthy adults. And chronic sleep restriction in animal models accelerates the formation of amyloid plaques and the spread of tau tangles. This provides a direct mechanistic link between poor sleep and the development of Alzheimer's disease. If you're not getting enough sleep, if you're not getting enough deep sleep, your brain is not efficiently clearing these toxic proteins, and they start to build up. Over time, this accumulation can reach a tipping point where it triggers a cascade of pathological processes that lead to neurodegeneration. Strongly supported, according to research on sleep deprivation and amyloid accumulation, published in PNAS and Science. But the story gets even more interesting. For a long time, researchers knew that the glymphatic system was more active during sleep, but they didn't fully understand the mechanism driving this increased activity. What was the engine powering this nightly cleaning process? A groundbreaking study published in the journal Cell in 2025 by Haugland and colleagues provided a remarkable answer. The driving force is synchronized, infraslow oscillations of the neurotransmitter norepinephrine. Norepinephrine is a neuromodulator that is typically associated with arousal, attention, and the stress response. It's released from a small cluster of neurons in the brainstem called the locus coeruleus. During wakefulness, norepinephrine levels are relatively high and variable, helping to keep you alert and responsive to your environment. But during NREM sleep, and particularly during slow-wave sleep, the activity of the locus coeruleus changes dramatically. Instead of the rapid, irregular firing seen during wakefulness, the locus coeruleus begins to fire in slow, rhythmic waves, with a period of about 50 seconds. These are infraslow oscillations, much slower than the typical brainwave frequencies we think of. Haugland and colleagues discovered that these slow waves of norepinephrine are the primary drivers of glymphatic clearance. Here's the mechanism. Norepinephrine acts on the smooth muscle cells that surround arteries in the brain, causing them to contract and relax in a rhythmic pattern called slow vasomotion. When norepinephrine levels surge, the arteries constrict, reducing cerebral blood volume. When norepinephrine levels drop, the arteries dilate and blood volume increases. This rhythmic change in blood volume is anti-correlated with CSF volume. When the arteries constrict and blood volume decreases, it creates space in the brain, and CSF rushes in to fill that space. When the arteries dilate and blood volume increases, CSF is pushed out. This creates a rhythmic pumping action, a pulsatile flow of CSF through the glymphatic pathways, effectively flushing out accumulated toxins. Strongly supported, according to the 2025 study by Haugland et al., published in Cell. This is an incredibly elegant mechanism. The brain is using the natural oscillations of a neurotransmitter system, synchronized with the slow rhythms of deep sleep, to drive a mechanical pumping process that cleans the brain. It's a beautiful example of how different levels of biological organization, neurochemistry, vascular dynamics, fluid mechanics, all come together to perform a critical function. But here's where this discovery has profound clinical implications. The same study by Haugland and colleagues showed that the common sleep aid zolpidem, which is sold under the brand name Ambien, actively suppresses these vital norepinephrine oscillations. Zolpidem is a GABA-A receptor agonist, meaning it enhances the activity of GABA, the brain's primary inhibitory neurotransmitter. It's very effective at inducing a state that looks like sleep on the surface. You lose consciousness, you're lying still, you're not responsive to the environment. But the sleep induced by zolpidem is not the same as natural sleep. In animal models, treatment with zolpidem reduced the amplitude of the norepinephrine waves by about 50%. And this reduction in norepinephrine oscillations had a direct, measurable impact on glymphatic function. The influx of a CSF tracer into the brain was decreased by more than 30% in zolpidem-treated animals compared to naturally sleeping animals. In other words, zolpidem was putting the animals to sleep, but it was simultaneously impairing the brain's ability to clean itself. Strongly supported, according to the 2025 study by Haugland et al., published in Cell. This finding raises serious concerns about the long-term use of certain sleep medications. If you're taking a drug that induces sleep but disrupts the underlying neurobiology required for the restorative functions of sleep, are you really getting the benefit you need? Are you actually protecting your brain, or are you potentially doing harm? By disrupting the precise neurochemical and vascular dynamics that power the glymphatic system, such medications may inadvertently impede the brain's ability to clear toxic proteins. And if this impairment is chronic, if it's happening night after night, year after year, it could theoretically contribute to the accumulation of beta amyloid and tau, and increase the risk for neurodegenerative diseases like Alzheimer's. Now, to be clear, this is an area of active research, and we don't yet have definitive long-term human data showing that chronic use of zolpidem directly causes Alzheimer's disease. But the mechanistic link is there, and it's concerning. It underscores a critical principle. The goal of sleep interventions should not be merely to induce unconsciousness. The goal should be to foster natural, biologically complete sleep with its intricate architecture and its underlying neurochemical dynamics intact. This is why behavioral and environmental strategies, things like maintaining a consistent sleep schedule, optimizing your sleep environment, managing light exposure, addressing stress, should always be the first line of defense for improving sleep. Pharmacological aids have their place, particularly for short-term use in specific clinical situations. But they should be used judiciously and under the guidance of a healthcare professional who understands these nuances. The discovery of the glymphatic system and the role of norepinephrine-mediated vasomotion has fundamentally changed our understanding of why sleep is so essential for brain health. Sleep is not just a time for your brain to rest. It's a time for your brain to actively clean itself, to flush out the toxic waste products that accumulate during waking hours. And this cleaning process is most efficient during deep, slow-wave sleep, driven by the rhythmic oscillations of norepinephrine and the pulsatile flow of cerebrospinal fluid. Protecting and prioritizing deep sleep is, quite literally, protecting your brain from the accumulation of the very proteins that drive neurodegeneration. The restorative symphony: memory, emotion, and metabolic health. We've talked about waste clearance, but that's just one piece of the restorative puzzle. Sleep actively facilitates a wide range of processes that are essential for cognitive function, emotional stability, and systemic health. Different stages of sleep appear to be specialized for distinct functions, and they work in concert throughout the night to consolidate memories, regulate emotions, and maintain metabolic and immune homeostasis. Let's start with memory. One of the most well-established and fascinating functions of sleep is its critical role in memory consolidation, the process by which fragile, short-term memories are transformed into stable, long-term representations. This is not a passive process where memories just sit there and solidify on their own. It's an active reorganization and strengthening of neural circuits, a process that requires specific patterns of brain activity that occur during sleep. NREM sleep, particularly the deep, slow-wave sleep of stage N3, is fundamentally important for the consolidation of declarative memories. Declarative memories are the memories you can consciously recall and describe. Factual knowledge like the capital of France or the formula for photosynthesis, and personal experiences like what you had for breakfast this morning or your last birthday party. These are sometimes called semantic and episodic memories, respectively. During slow-wave sleep, the brain engages in a process called systems consolidation. This involves a coordinated neural dialogue between the hippocampus, which is a seahorse-shaped structure deep in the brain that is critical for forming new memories, and the neocortex, the outer layer of the brain where long-term memories are ultimately stored. The slow oscillations that characterize N3 sleep are thought to orchestrate the reactivation or replay of neural activity patterns that were associated with recent experiences. Essentially, the hippocampus is replaying the events of the day, and the neocortex is listening, gradually integrating these new memories into its existing networks. Over time, with repeated reactivation during multiple nights of sleep, the memory trace is transferred from the hippocampus to the cortex, where it becomes more stable and less dependent on the hippocampus for retrieval. Strongly supported, according to research on sleep and memory consolidation published in PMC and Sleep Medicine Clinics. This is why a good night's sleep after learning something new is so important. If you study for an exam and then stay up all night, you're depriving your brain of the opportunity to consolidate that information. The memories remain fragile, and you're much more likely to forget them. But if you study and then get a full night of sleep, especially sleep rich in slow-wave activity, those memories are strengthened and integrated, and you're much more likely to retain them over the long term. REM sleep, on the other hand, appears to be more critical for the consolidation of procedural memories. Procedural memories are the how-to memories, the skills and habits that you learn through practice. Riding a bike, playing a musical instrument, typing on a keyboard, performing a complex motor sequence. These are often implicit memories, things you can do without consciously thinking about the steps involved. The unique neurochemical environment of REM sleep, with its high levels of acetylcholine and low levels of norepinephrine, is believed to promote synaptic plasticity, the ability of connections between neurons to strengthen or weaken. This plasticity is essential for refining motor skills and integrating new procedures into existing motor programs. Studies have shown that people who are deprived of REM sleep after learning a new motor task show impaired performance on that task the next day, while those who get adequate REM sleep show significant improvement. REM sleep is also thought to play a role in integrating emotional information into procedural memories, which may be why emotionally charged experiences are often remembered more vividly and can influence our future behavior more strongly. Strongly supported, according to research on REM sleep and procedural memory published in PMC and Sleep Medicine Clinics. But sleep's role in emotional regulation goes far beyond just memory. Sleep is profoundly intertwined with your ability to manage your emotions, to respond appropriately to emotional stimuli, and to maintain emotional stability. A lack of sleep significantly disrupts these processes, often leading to heightened irritability, mood swings, increased reactivity to negative stimuli, and a reduced ability to experience positive emotions. Neuroimaging studies have revealed the neural basis for this. Sleep deprivation disrupts the functional connectivity between the amygdala, which is the brain's primary emotion processing center, and the prefrontal cortex, or PFC, which is the part of the brain responsible for executive functions, like planning, decision-making, and emotional regulation. The prefrontal cortex exerts top-down control over the amygdala, helping to contextualize emotional responses and inhibit inappropriate or excessive reactions. In a well-rested brain, when you encounter something emotionally provocative, your amygdala responds, but your prefrontal cortex steps in to modulate that response, to put it in perspective, to help you respond in a measured and appropriate way. But in a sleep-deprived brain, this system breaks down. The amygdala becomes hyper-reactive, generating exaggerated emotional responses, while the prefrontal cortex becomes less active and less effective at regulating those responses. The result is that you're more likely to overreact to minor frustrations, to feel overwhelmed by stress, to experience intense negative emotions, and to have difficulty controlling your impulses. Strongly supported, according to research on sleep deprivation and emotional regulation published in PMC, REM sleep, in particular, plays a crucial role in emotional homeostasis. One leading theory proposed by researchers like Matthew Walker suggests that REM sleep helps to divorce the emotional charge from the memory of an experience. During a REM, the brain reprocesses emotional events in a neurochemically safe environment, characterized by low levels of norepinephrine, which is the brain's primary stress neurotransmitter. By replaying emotional memories in this low norepinephrine state, the brain can consolidate the factual details of the event while reducing the visceral, emotional intensity associated with it. This allows you to remember what happened without being overwhelmed by the emotion every time you recall it. It's a form of emotional therapy that happens naturally every night, helping you to build emotional resilience and to move forward from difficult experiences. Preliminary evidence. This is a leading theory supported by some research, but the exact mechanisms are still being investigated. Chronic disruption of sleep, especially REM sleep, is a common feature of and a significant risk factor for mood disorders such as depression and anxiety. In fact, insomnia and other sleep disturbances often precede the onset of these disorders, suggesting that poor sleep may not just be a symptom, but a contributing cause. Improving sleep quality can be a powerful intervention for improving mood and emotional well-being. Beyond the brain, sleep is also a powerful regulator of metabolic and immune health. Deep NREM sleep is a key period for hormonal regulation. As we mentioned earlier, it triggers the most significant daily release of growth hormone, which is essential for cellular repair, bone density, and maintaining lean body mass. But sleep also helps regulate the circadian rhythm of cortisol, the body's primary stress hormone. Cortisol levels should be low at night and rise in the early morning to help you wake up. But sleep disruption can lead to elevated cortisol levels at night, which is associated with insulin resistance, increased appetite, and metabolic dysfunction. Indeed, insufficient sleep is a major risk factor for metabolic disorders. Even partial sleep restriction, getting just 5 or 6 hours of sleep per night instead of the recommended 7 to 9, can significantly reduce insulin sensitivity, impairing your body's ability to regulate blood glucose. This increases the risk of developing type 2 diabetes. This metabolic impairment is partly due to hormonal dysregulation. Sleep deprivation increases levels of the hunger hormone ghrelin and decreases levels of the satiety hormone leptin, making you feel hungrier and less satisfied after eating. But it's also because sleep deprivation promotes a state of low-grade systemic inflammation, strongly supported, according to research on sleep and metabolic health published in PMC and Diabetology and Metabolic Syndrome. During sleep deprivation, the body increases its production of pro-inflammatory cytokines, such as interleukin-6 or IL-6 and tumor necrosis factor or TNF. These are signaling molecules that are part of the immune response, and they're useful in the short term for fighting infections. But when they're chronically elevated, they create a state of low-grade inflammation that contributes to insulin resistance, promotes fat storage, damages blood vessels, and weakens immune function. Chronic inflammation is a common underlying factor in many chronic diseases, including heart disease, diabetes, obesity, and even cancer. Sleep is also critical for immune function. During sleep, your body ramps up the production of immune cells and antibodies. Studies have shown that people who are sleep-deprived are more susceptible to infections. If you expose sleep-deprived individuals to a cold virus, they're significantly more likely to get sick compared to well-rested individuals. And if you get a vaccine while you're sleep-deprived, you may produce fewer antibodies in response, meaning the vaccine is less effective. Sleep is when your immune system is doing some of its most important work, building up your defenses and preparing to fight off pathogens. Strongly supported, according to research on sleep and immune function published in PMC. So you can see that sleep is not just about the brain. It's a whole body phenomenon. It's when your brain consolidates memories and processes emotions, when your body repairs tissues and regulates hormones, when your immune system strengthens its defenses, and when your metabolic systems recalibrate. Sleep is a critical pillar of health, actively working to sharpen your mind, balance your emotions, and fortify your body's defenses against disease. Sleep across the lifespan, aging and the decline of deep sleep. Sleep is not static across the human lifespan. The way we sleep changes dramatically from infancy through old age, and these changes have profound implications for health, particularly for cognitive health in later life. Understanding how sleep architecture evolves as we age can help us appreciate the importance of protecting sleep quality throughout our lives, and can inform strategies for mitigating age-related cognitive decline. In infancy, sleep is dominated by a precursor to REM sleep, sometimes called active sleep. Newborns spend about 50% of their sleep time in this state, which is thought to be vital for the intense neural development occurring during this critical period. The brain is forming trillions of synaptic connections, building the foundational architecture of the nervous system, and this process appears to require the unique neurochemical environment and high levels of brain activity characteristic of REM sleep. As we move through childhood and into adolescence, sleep patterns shift. Total sleep time gradually decreases and the proportion of REM sleep declines to adult levels, around 20 to 25% of total sleep. But adolescence also brings a shift in the timing of sleep. a biological phenomenon known as a delayed sleep phase. Teenagers naturally want to go to bed later and wake up later. This is not laziness or poor discipline, it's a genuine shift in their circadian rhythm, driven by changes in the sensitivity of the circadian system to light and changes in the homeostatic sleep drive. Unfortunately, this biological shift often clashes with early school start times, leading to chronic sleep deprivation in many adolescents, which can have significant consequences for their academic performance, mood, and physical health. Strongly supported, according to research on sleep across the lifespan. Entering adulthood, sleep patterns stabilize. Most adults need 7 to 9 hours of sleep per night, and the architecture of sleep, the proportions of N1, N2, N3, and REM, settles into a relatively consistent pattern. But as we progress into middle age and older adulthood, sleep quality and structure begin to decline. And one of the most consistent and consequential changes is a dramatic reduction in the amount of deep, slow-wave sleep, N3. Studies have shown that the percentage of N3 sleep decreases by approximately 2% per decade of life, starting in early adulthood. By the time you reach your 60s or 70s, you may be getting only a fraction of the deep sleep you got in your 20s. This decline often plateaus after the age of 60, but the damage, in a sense, has already been done. The decline is often more pronounced in men compared to women, though both sexes experience it. Concurrently, older adults tend to experience an increase in lighter N1 and N2 sleep, as well as more frequent awakenings during the night, a condition known as increased wake after sleep onset, or WASO. This leads to more fragmented and less restorative sleep. Strongly supported, according to research on aging and slow-wave sleep published in JAMA Neurology and PMC. These changes are paralleled by age-related hormonal shifts. The decline in slow-wave sleep is tightly linked to a decline in growth hormone secretion, which is normally released during N3. Older adults also tend to have higher nocturnal cortisol levels, which is associated with sleep fragmentation and can contribute to metabolic dysfunction and cognitive impairment. Now, you might think, well, that's just a normal part of aging. What can you do? But here's the critical point. This age-related decline in slow-wave sleep is not a benign phenomenon. It's increasingly recognized as a significant risk factor for cognitive decline and neurodegenerative diseases, most notably Alzheimer's disease. And the link is believed to be mediated in large part by the glymphatic system. As we discussed earlier, glymphatic clearance of neurotoxic waste products like beta amyloid is most efficient during slow-wave sleep. Therefore, the age-related loss of slow-wave sleep directly translates to a reduction in the brain's nightly cleaning efficiency. You're getting less of the very stage of sleep that is most important for clearing out the toxic proteins that drive neurodegeneration. And compounding this issue, the integrity of the glymphatic system itself appears to degrade with age. Animal studies have shown a functional decline of the glymphatic system of up to 80 to 90% in aged brains compared to young ones. So you have a double hit: less slow-wave sleep and a less efficient clearance system. This combination creates a perfect storm for the accumulation of toxic proteins. Strongly supported, according to research on aging, glymphatic function, and dementia risk published in PMC and Nature Communications. One particularly striking prospective cohort study, published in JAMA Neurology, followed older adults over a period of 17 years and measured their sleep architecture. The researchers found that for each percentage point decrease in slow-wave sleep per year, there was an associated 27% increase in the risk of developing dementia over the follow-up period. That's a substantial association. It suggests that the loss of deep sleep is not just a marker of aging, but a potentially modifiable risk factor for dementia. Strongly supported, according to the study published in JAMA Neurology. The relationship between sleep disruption and dementia is often described as bidirectional and cyclical, a vicious cycle. Poor sleep, particularly the loss of slow-wave sleep, impairs glymphatic clearance, leading to the accumulation of beta-amyloid and tau proteins. These protein aggregates are toxic to neurons. They can damage the very brain regions that are critical for generating deep sleep and regulating the sleep-wake cycle, such as the ventrolateral preoptic area, the locus coeruleus, and other brainstem nuclei. This damage further disrupts sleep, which accelerates protein accumulation, which causes more neuronal damage, which disrupts sleep even more. It's a self-perpetuating cycle that drives disease progression. While the loss of slow-wave sleep has been a major focus, some research also points to changes in REM sleep as being associated with dementia risk. Some studies have found that a lower percentage of REM sleep or a longer delay to the first REM period is associated with a higher risk of incident dementia. The overall picture is complex, and different aspects of sleep architecture may contribute to risk in different ways. But the evidence strongly suggests that the deterioration of sleep quality and architecture with age is a key and potentially modifiable risk factor in the pathogenesis of dementia. Preliminary evidence. The relationship between REM sleep changes and dementia. Risk is an area of ongoing research. This positions the preservation and improvement of sleep in older adults as a critical strategy for promoting brain health and potentially delaying or preventing the onset of neurodegenerative disease. It's not inevitable that your sleep has to deteriorate dramatically as you age. There are things you can do to protect sleep quality: maintaining a consistent sleep schedule, getting regular physical activity, managing stress, optimizing your sleep environment, treating sleep disorders like sleep apnea, and being cautious about medications that can disrupt sleep architecture. These are not trivial interventions. They may be some of the most important things you can do to protect your brain as you age. The consequences of sleep disruption, a path to neurodegeneration. We've touched on this throughout, but let's bring it all together and talk explicitly about the consequences of insufficient or disordered sleep. The effects extend far beyond simple daytime tiredness. Chronic sleep disruption constitutes a significant public health concern with profound repercussions for neurological and systemic health. Sleep deprivation, whether acute or chronic, triggers a cascade of detrimental physiological processes that can accelerate cognitive decline and increase the risk for a host of diseases, particularly neurodegenerative disorders like Alzheimer's and Parkinson's disease. The relationship is often bidirectional, as we've discussed. Sleep disturbances act as both a cause and a consequence of the underlying pathology, creating a self-perpetuating cycle of neurodegeneration. Let's walk through the key mechanisms linking sleep loss to brain disease. First and foremost is the impairment of the glymphatic waste clearance system. We've spent a lot of time on this, and for good reason. The glymphatic system is most active during deep sleep. Any reduction in sleep duration or quality compromises the brain's ability to flush out toxic metabolic byproducts. This leads to the accumulation of proteins like beta-amyloid, tau, and alpha-synuclein. Even a single night of sleep deprivation has been shown to increase beta-amyloid levels in the cerebrospinal fluid of healthy adults. And chronic sleep restriction in animal models accelerates the formation of amyloid plaques and the spread of pathological tau tangles. Similarly, sleep loss impairs the clearance of alpha-synuclein, the protein that aggregates to form Lewy bodies in Parkinson's disease and other synucleinopathies. Strongly supported, according to research on sleep deprivation and protein accumulation published in PNAS Science and PMC. Beyond impaired waste clearance, sleep deprivation promotes a state of heightened neuroinflammation and oxidative stress. Insufficient sleep leads to the excessive activation of microglia, the brain's resident immune cells. Microglia play a protective role in clearing debris and fighting infections, but when they're chronically activated, they become a source of damage. They release a barrage of pro-inflammatory cytokines and reactive oxygen species that can damage neurons, disrupt synaptic function, and exacerbate protein pathology. This low-grade chronic neuroinflammation is a common feature across all major neurodegenerative diseases. It's not just a consequence of the disease, it's an active driver of disease progression. Strongly supported, according to research on sleep deprivation and neuroinflammation published in PMC and Frontiers TUS. Furthermore, sleep loss disrupts the brain's delicate balance between the production of damaging reactive oxygen species, which are a byproduct of normal metabolism, and the antioxidant systems that neutralize them. The resulting increase in oxidative stress can lead to widespread damage to lipids, proteins, and DNA, contributing to neuronal dysfunction and death. Sleep is a time when antioxidant defenses are upregulated and when the brain can repair oxidative damage. Without adequate sleep, this balance is lost. Disruption of circadian rhythms, which often accompanies poor sleep habits and is a core feature of neurodegenerative diseases, further compounds the damage. The molecular clocks that regulate cellular function are critical for everything from DNA repair to metabolic efficiency to the timing of gene expression. When these rhythms are desynchronized from the external environment or from each other, cellular processes become disorganized. This disruption can worsen protein pathology, impair mitochondrial function, and lead to the loss of synapses and neurons in critical brain regions. Strongly supported, according to research on circadian disruption and neurodegeneration published in PMC and Nature Aging. The link between sleep disorders and neurodegeneration is starkly illustrated by a condition called REM, sleep behavior disorder, or RBD. In RBD, individuals physically act out their dreams due to a failure of the normal muscle atonia that should occur during REM sleep. They might punch, kick, shout, or even get out of bed and engage in complex behaviors, all while still asleep. This can be dangerous for both the individual and their bed partner. But what's truly remarkable about RBD is that it's now recognized as a powerful early predictor of neurodegeneration. Over 80% of individuals diagnosed with RBD eventually develop a synucleinopathy, like Parkinson's disease, dementia with Lewy bodies, or multiple system atrophy, often years or even decades after the sleep disorder first appears. RBD is thought to reflect early pathology in the brainstem regions that control REM sleep and muscle atonia, regions that are also affected early in the course of synucleinopathies. This highlights that sleep disturbances are not merely a symptom that appears late in the disease process. They can be part of the earliest stages of the disease itself, offering a potential window for early detection and intervention. Strongly supported, according to research on REM sleep behavior disorder, Neurodegeneration published in Scientific American and PMC. The cumulative evidence firmly establishes that chronic sleep disruption is a potent and modifiable risk factor that actively drives the fundamental pathological mechanisms of neurodegeneration. It's not just that people with Alzheimer's or Parkinson's tend to have poor sleep. It's that poor sleep, sustained over years and decades, can contribute to the development of these diseases in the first place. This is a critical point because it means that improving sleep is not just about feeling better in the short term. It's a long-term investment in brain health, a way to reduce your risk for some of the most devastating diseases of aging. Practical wisdom, sleep hygiene, chronotypes, and building better sleep, given everything we've discussed, the intricate regulatory systems, the structured architecture of sleep stages, the critical role of the glymphatic system, the profound impact on memory, emotion, and metabolic health, and the link between poor sleep and neurodegeneration, it's clear that adopting strategies to improve sleep quality and duration is not optional. It's a critical component of a healthy lifestyle, on par with nutrition and exercise. But here's the thing, sleep is not one size fits all. We all have unique biological predispositions, unique internal rhythms, and unique challenges. A personalized approach that considers your individual chronotype, your lifestyle, and your specific sleep issues can be far more effective than generic advice. So let's talk about practical strategies, starting with the concept of chronotype. Your chronotype refers to your innate preference for the timing of sleep and wakefulness, essentially your internal biological clock's natural tendency. Some people are naturally early risers, feeling most alert and productive in the morning. Others are night owls, feeling most energized and creative in the evening. And many people fall somewhere in between. These preferences are often categorized into archetypes, and one popular framework uses animal names. Lions are the early morning types. Wolves are the late evening types. Bears follow the sun's cycle and represent the majority of the population. And dolphins are light, easily disturbed sleepers who often struggle with insomnia. Your chronotype is influenced by genetics, age, and environmental factors, and it has real physiological underpinnings related to the phase of your circadian rhythm. Understanding and working with your chronotype rather than against it can significantly improve your sleep quality and daytime function. For example, if you're a wolf, a late chronotype, and you're forced into an early morning work schedule, you're experiencing what we talked about earlier: social jet lag, a chronic misalignment between your internal clock and your external schedule. This can lead to chronic sleep deprivation, poor performance, mood disturbances, and long-term health consequences. Strongly supported, according to research on chronotypes published in Sleep Foundation and BMJ Public Health. Where possible, adjusting your work or social schedules to better match your chronotype can be highly beneficial. If you're a wolf and you have the flexibility to start work later in the day, that alignment can make a huge difference. But for many people, that kind of flexibility isn't an option. If you're stuck with a schedule that doesn't match your chronotype, there are still strategies you can use to help shift your internal clock and improve alignment. For an evening type who needs to wake up earlier, maximizing bright light exposure immediately upon waking can help shift your circadian rhythm earlier, making it easier to fall asleep earlier the next night. Conversely, if you're a morning type who needs to stay up later, getting bright light exposure in the evening can help delay your rhythm. Beyond chronotype-specific adjustments, there's a set of universal practices known as sleep hygiene that can form the foundation for better sleep. These are evidence-based strategies that help to optimize the conditions for sleep and to reinforce your natural sleep-wake rhythms. The single most important element of sleep hygiene is maintaining a consistent sleep-wake schedule. Go to bed and wake up at approximately the same time every day, even on weekends. This consistency is crucial for reinforcing your circadian rhythm. Your SCN, Your master clock thrives on predictability. When you go to bed and wake up at the same time every day, you're giving your SCN a clear, consistent signal, and it can entrain your entire physiology to that rhythm. This makes it easier to fall asleep at night and easier to wake up in the morning feeling refreshed. Conversely, if your sleep schedule is erratic, if you're going to bed at midnight one night and 3 a.m. the next, your SCN is getting conflicting signals and your circadian rhythm becomes desynchronized. This makes sleep more difficult and less restorative. Strongly supported, according to research on sleep hygiene published in MDPY and Yale School of Medicine, the strategic management of light exposure is also crucial. Light is the most powerful Zeitgeber for your circadian system. Maximizing exposure to bright, natural light during the day, especially in the morning, helps to anchor your circadian rhythm and promote daytime alertness. If you can, spend time outdoors in the morning or at least near a window where you're exposed to natural light. This sends a strong signal to your SCN that it's daytime, reinforcing wakefulness and helping to set your internal clock. Conversely, it's vital to minimize exposure to bright light, particularly blue wavelength light, in the 1 to 2 hours before bedtime. Blue light, which is emitted in high amounts by electronic screens like smartphones, tablets, computers, and televisions, is particularly effective at suppressing melatonin production. When you're staring at a bright screen late at night, you're sending a signal to your SCN that it's still daytime, that it's not yet time to prepare for sleep. This delays the onset of melatonin secretion and makes it harder to fall asleep at your desired time. If you must use screens in the evening, consider using blue light filters or apps that shift the color temperature of your screen to warmer tones. Better yet, try to establish a screen-free wind-down routine in the hour or two before bed, engaging in relaxing activities like reading a physical book, taking a warm bath, or practicing gentle stretching or meditation. Creating an optimal sleep environment is another key factor. Your bedroom should be a sanctuary for sleep, a place that is conducive to rest. This means keeping it dark, quiet, and cool. Darkness is important because even small amounts of light can suppress melatonin production and disrupt sleep. Use blackout curtains or an eye mask if necessary. Quiet is important because noise can fragment sleep, causing brief arousals that you may not even remember, but that still disrupt the continuity of your sleep cycles. Use earplugs or a white noise machine if you live in a noisy environment. And cool is important because a drop in core body temperature is a natural signal for sleep. An ambient temperature of around 18 to 20 degrees Celsius or 65 to 68 degrees Fahrenheit is often recommended. If your bedroom is too warm, it can interfere with this natural temperature drop and make it harder to fall asleep and stay asleep. The timing of meals, exercise, and substance use also has a significant impact on sleep. Avoid heavy meals in the 2 to 3 hours before bedtime. Digestion is an active process that can interfere with sleep, onset, and quality. Similarly, avoid vigorous exercise in the late evening. While regular physical activity is excellent for sleep in general, exercising too close to bedtime can be stimulating and make it harder to fall asleep. Aim to finish vigorous workouts at least 3 to 4 hours before bed. Caffeine is a powerful stimulant that blocks adenosine receptors, effectively masking your sleep pressure. It has a half-life of about 5 to 6 hours, meaning that half of the caffeine you consume is still in your system 5 to 6 hours later. If you have a cup of coffee at 4 p.m., a significant amount of caffeine is still circulating in your bloodstream at 10 p.m., potentially interfering with your ability to fall asleep. For most people, it's best to avoid caffeine in the afternoon and evening. Strongly supported, according to research on caffeine and sleep published in PMC. Alcohol is another substance that many people mistakenly believe helps with sleep. While alcohol can induce drowsiness and help you fall asleep faster, it significantly disrupts sleep quality. It fragments sleep in the second half of the night, causing more awakenings, and it suppresses REM sleep, depriving you of that critical stage for absorbed, emotional processing and memory consolidation. The sleep you get after drinking alcohol is not restorative sleep. It's better to avoid alcohol in the hours before bedtime if you want to optimize your sleep quality. When lifestyle modifications are insufficient, when you've tried all the behavioral and environmental strategies, and you're still struggling with sleep, it can be tempting to turn to pharmacological interventions. And there are certainly situations where sleep medications can be helpful, particularly for short-term use in specific clinical contexts, such as acute insomnia triggered by a stressful life event. But these medications must be approached with caution and with a clear understanding of their limitations and potential risks. As we discussed in detail earlier, not all sleep is created equal. The common sleep aid zolpidem, for instance, is very effective at inducing a state that looks like sleep on the surface, but it disrupts the underlying neurobiology required for sleep's restorative functions. Specifically, it suppresses the slow, rhythmic waves of norepinephrine that drive the glymphatic system's waste clearance function. By interfering with this natural process, the medication may promote a form of sleep that is less effective at cleaning the brain of neurotoxic waste. This is not to say that zolpidem or other sleep aids are never appropriate, but it underscores a critical principle. The goal should not be merely to induce unconsciousness. The goal should be to foster natural, biologically complete sleep with its intricate architecture and its underlying neurochemical dynamics intact. This is why behavioral and environmental strategies should always be the first line of defense for improving sleep. Cognitive behavioral therapy for insomnia, or CBTI, is a structured, evidence-based approach that addresses the thoughts, behaviors, and environmental factors that contribute to poor sleep. It's been shown to be highly effective, often more effective than medication in the long term, and it doesn't carry the risks of side effects or dependency. If you're struggling with chronic insomnia or other sleep issues, seeking out a qualified sleep specialist or a therapist trained in CBT can be a very worthwhile investment. The overarching message here is that sleep is not a luxury. It's a biological necessity. And protecting your sleep, prioritizing it, treating it with the same level of importance that you give to diet and exercise is one of the most powerful things you can do for your health. It's an investment in your cognitive function, your emotional well-being, your metabolic health, your immune resilience, and your long-term brain health. It's an investment in your ability to think clearly, to feel good, to perform at your best, and to reduce your risk for some of the most devastating diseases of aging. Bringing it all together, sleep as a foundation for life. So let's take a step back and bring all of this together. We've journeyed deep into the hidden biology of sleep, exploring the elegant regulatory systems that govern when you feel sleepy and when you feel alert, the intricate architecture of sleep stages that cycle throughout the night, the master clocks and peripheral timekeepers that orchestrate your body's daily rhythms, and the remarkable glymphatic system that cleans your brain while you sleep. We've seen that sleep is not a passive state, not a simple shutdown, but an extraordinarily active and meticulously orchestrated biological process. It's when your brain consolidates memories, transferring fragile short-term traces into stable, long-term storage. It's when your brain processes emotions, helping you to regulate your responses and build emotional resilience. It's when your brain quite literally cleans itself, flushing out toxic waste products like beta amyloid and tau that, if allowed to accumulate, can drive neurodegeneration. It's when your body repairs tissues, releases growth, hormone, reinforces your immune system, and recalibrates your metabolic processes. We've learned that the quality and structure of your sleep matter just as much as the quantity. You need to move through complete, uninterrupted cycles, experiencing the full spectrum of sleep stages in their natural proportions. You need the deep, slow-wave sleep of N3 for physical restoration and waste clearance. You need the REM sleep for emotional processing and memory consolidation. And you need these stages to occur at the right times, aligned with your circadian rhythm and with the external cycle of day and night. We've explored how sleep changes across the lifespan and how the age-related decline in slow-wave sleep is not a benign phenomenon, but a significant risk factor for cognitive decline and dementia. We've seen how chronic sleep disruption can trigger a cascade of detrimental processes: impaired glymphatic clearance, neuroinflammation, oxidative stress, circadian desynchrony that actively drive the pathological mechanisms of neurodegeneration. And we've talked about practical strategies for protecting and improving your sleep, understanding and working with your chronotype, maintaining a consistent sleep schedule, managing light exposure, creating an optimal sleep environment, being mindful of the timing of meals and exercise and substance use, and prioritizing behavioral interventions over pharmacological ones whenever possible. The evidence is overwhelming. Healthy sleep is a cornerstone of neurological health and overall well-being. It's not something to be sacrificed in the pursuit of productivity or entertainment. It's not a sign of weakness or laziness. It's a fundamental biological imperative, as essential to your health as the food you eat and the air you breathe. And in our modern world where artificial light, shift work, electronic screens, and a culture that often glorifies busyness and sleep deprivation are ubiquitous, protecting your sleep requires conscious effort and intentionality. But the payoff is immense. When you prioritize sleep, when you give your brain and body the time and the conditions they need to perform their nightly restorative work, you're setting yourself up for success in every domain of your life. You think more clearly, you learn more effectively, you remember more accurately, you regulate your emotions more skillfully, you're more resilient to stress, you're more creative, you're healthier with a stronger immune system, better metabolic function, and a lower risk for chronic diseases. And you're protecting your brain for the long term, reducing your risk for cognitive decline and neurodegenerative diseases like Alzheimer's and Parkinson's. Sleep is quite literally an investment in your future self. Every night of good sleep is a deposit into your health bank account, building up reserves of cognitive resilience, emotional stability, and physical vitality that will serve you for years and decades to come. And conversely, every night of poor sleep, every night of insufficient sleep, is a withdrawal from that account, a small but cumulative deficit that can add up over time to significant consequences. So, I encourage you to take this information, to really absorb it, and to think about your own sleep. Are you giving it the priority it deserves? Are you creating the conditions for good sleep? Are you listening to your body's signals, respecting your natural chronotype, and aligning your schedule with your circadian rhythm as much as possible? Are there changes you could make, small adjustments to your routine or your environment that could improve your sleep quality? You don't have to be perfect. Life is complicated. And there will be times when sleep is disrupted by circumstances beyond your control. But if you can make sleep a priority, if you can protect it and nurture it as a fundamental pillar of your health, you'll be doing one of the most important things you can do for your brain, your body, and your overall quality of life. Sleep is not a mystery anymore. We understand in remarkable detail what happens during those hours of darkness, why it matters so profoundly, and how to optimize it. The hidden biology of sleep is no longer hidden. It's a story of elegant regulatory systems, of intricate neural architecture, of molecular clocks and waste clearance networks, of memory consolidation and emotional processing, of hormonal regulation and immune function. It's a story of how your brain and body work together, night after night, to keep you healthy, sharp, and resilient. And it's a story that, I hope, inspires you to treat your sleep with the respect and the care it deserves. Because when you sleep well, you live well, you think well, you feel well, and you're building a foundation for a long, healthy, cognitively vibrant life. If you found this deep dive into the biology of sleep valuable, if it gave you a new appreciation for what's happening in your brain and body every night, I'd really appreciate it if you could take a moment to like this video and subscribe to the channel. Your support helps me continue to create this kind of in-depth, science-based content on topics like sleep, metabolic health, fasting, and optimal living. And if there are other topics you'd like me to explore in this level of detail, please let me know in the comments. I read them all, and your suggestions help guide the direction of future videos. Thank you so much for spending this time with me, for engaging with this material, and for prioritizing your health and your understanding of how your body works. I wish you restful, restorative sleep and vibrant health. Take care, and I'll see you in the next video.